When do you want to start?
Yeah, start. Yeah. Everybody, at least in the middle of the back.
Good morning, everybody. Welcome to Grand Rounds, and welcome to Grand Rounds at the distant sites. This morning's presenter is Dr. Val Montessori. He's a member of the Division of Infectious Diseases and one of the former chief residents in medicine at St. Paul's just a few years ago, who's now the proud owner of a Labradoodle that I think we're going to see. So, Dr. Montessori.
First, can you guys hear me? Yes. Okay, good. Did you hear my whole conversation about the dog, too? Okay, good. Phew. Okay. So, thank you for having me. I always think when I'm going to do grand rounds, like, what do people actually want to hear about that's not MRSA? So I spent a bit of time thinking about it, and I wanted to talk about something that was a little bit interesting and topical. So I dove into this idea of the microbiome, and that is a slippery slope down a rabbit hole, people. I was in there, and I came out 2 months later, and here I am. And so I'm going to try and give you the condensed view of what I got out of it, and mostly focus on what I realized was interesting to me as a clinician, because there's a lot of stuff out there that is really of interest to basic scientists and people who do epidemiological work and things like that. But as a clinician, really what I focused on was things that I thought you guys might like. So I'm going to describe to you the healthy human gastrointestinal microbiome as far as we know it today, and then focus on what we know about the effect of antibiotic therapy on the gastrointestinal microbiome, and then a very few clinical associations that people have described with changes in the microbiome. And I'm saying very few because the list is very, very long. And then I'll go through a couple of clinical studies of therapeutic manipulations of the GI microbiome. Because like I said, I'm a clinician, and I thought, okay, well, that's all very interesting, but what does it mean? So I'm going to talk particularly about fecal transplant and then about probiotics, which is super fun. So I made this slide about conflict of interest, and I really meant financial conflict of interest because, first of all, ID people don't make a lot of money, so we don't have a lot of financial conflict of interest. But aside from that, then I realized I actually do have some disclaimers to make. One is that I am not a lab medicine person. I'm not a microbiologist. I'm not a molecular biochemist. And I'm certainly not a basic scientist. So if you ask me questions like, what primers did they use and what cycler did they use and things like that, I might cry a little bit and run away. So don't ask me that stuff. I will give you my vague understanding of things and then try and tie it to clinical medicine. Okay, so what does it mean, the human microbiome? 10 years ago, I don't think I'd ever even heard that word. So it's a new word that tries to describe the concept that in healthy humans, we have a heck of a lot of bacteria hanging around, and that actually bacterial cells far, far, far outnumber human cells. And about 10 years ago, the NIH set out to try and describe what microorganisms are present in the healthy human host and try and give some idea of the concept that the identity and the degree of diversity of these bacteria vary from person to person, and they vary in different disease states. But up until about 10 years ago, we really had very little idea of what that consisted of. And in fact, some very basic numbers are that we believe that microorganisms outnumber human cells by a ratio of 10 to 1. So you're like, oh, okay, that's not a very big number. But their genes outnumber human genes by a factor of at least 200. And this is the one that I like because, you know, women always focus on these stupid things like weight. But the microbiome mass accounts for up to 5 pounds of body weight. So really, I'm 5 pounds lighter than I think I am. So what is this Human Microbiome Project? We've all kind of heard about it, but what is it? Why? How? What's that whole story? So as I said, it dates back to about 10 years ago when the NIH wanted to focus on characterizing the human microbiome and analyze its role in health and disease. Sounds very straightforward. And the main reason is that they realized that we have a basic understanding that bacteria are associated with infections. I mean, that's what I've spent my whole medical career focusing on. But we also have this, well, at least we recognize that bacteria are also good. They colonize surfaces on the insides of the body and are essential for life. And most of us in medical school sort of had some inkling that bacteria help to digest food, that they might help in the synthesis of vitamin K and B12. But over the last 10 years, there's been increasing interest in other functions of the bacteria. And the one that you will hear more and more as we talk this afternoon, is how the bacteria and their fermentation products or their metabolic products affect our body. And the one that, like I said, you might sort of hear popping up is this whole concept of short-chain fatty acids. Now, have any of you thought of short-chain fatty acids any time in the last 10 years unless you were in medical school? No, right? I'm like, what? Okay, so it's a big thing in this whole concept of the human gut microbiome and how it affects our health. But it seems that short-chain fatty acids are produced when the bacteria ferment dietary fiber, so non-absorbed dietary fiber. It's the main thing that they produce. And some of them are very important to just the health of the intestinal epithelial cells. So right there's this idea that the more fiber you eat, the more short-chain fatty acids are produced, the healthier your gut intestinal epithelia might be. Fine. But it also seems that short-chain fatty acids can and are able to adhere to receptors that are involved in a whole bunch of other functions, including endocrine functions, immune cells. And so there's this concept that maybe they are involved in modulating the central nervous system. There's also the realization that metabolism of tryptophan in the gut is related to central nervous system function through production of serotonin. So there's a very, very complex situation that's becoming increasingly studied about not just what the bacteria are, but how their life, how they're functioning, how their metabolism is directly influencing your metabolism and your health. That's really hard, right? Short-chain fatty acid. I'm like, what? Okay, so in fact, many of the people who are involved in this type of work have stopped thinking about humans and microorganisms, but they talk about the human microbiota supraorganism, that we are in fact made of human cells and microorganism cells, and that we exist in this symbiotic relationship where, they make some metabolites that are beneficial to us, we provide them with undigested fiber and a nice place to live. So it's a kind of a back and forth situation, and really we are not just human, but we are human with microorganisms, which kind of makes me very, very happy right here. So what was the Human Microbiome Project, and how did they try and identify which bacteria were important in our health? Well, apparently they looked at 5 body areas. They looked at the skin, the mouth, the nose, the colon, and the vagina. And then they used the specific molecular biology techniques to sequence DNA, and by DNA there may have also been RNA, really don't ask me those hard questions, of organisms, and tried to match them to all sorts of computer logs of known organisms. And the part that kind of surprised me when I started looking at this, and maybe you guys all knew this, but most of the organisms that they talk about when they talk about the gut microbiome in the Microbiome project cannot be cultured in the regular lab. If you think about it, a large proportion of them are anaerobes, and really at the best of times we're not that good at culturing anaerobes, so it's not really that surprising, but I thought it was interesting to notice that it's not a matter of just taking a little lump of stool and then taking it to the lab and culturing it. Really, it's a very complicated formula and technique. So as I said, multiple body sites, a lot of specimens, multiple reference strains, and you can imagine if you're starting to look at all these genes, this must have taken them a super-de-duper long time. And as I said, most of them are not culturable, and really you end up with a whole lot of information about a whole lot of organisms. So what kind of information can we synthesize from what they've tried to identify and describe? First of all, when I started reading about these things, I realized that I had a little panic attack because I thought the microbiologists were playing a trick again and changing names again. Because here I am, been doing ID for a while, and the first two words that came up over and over again were this firmicutes or firmicutes and Bacteroidetes. And Bacteroidetes, I'm like, okay, I think I kind of know what that is. It sounds like Bacteroides. I think I maybe could figure that one out. But this one, never heard of it. Never seen it. And I thought, great, they're trying to mess with me. But then I realized that really when the microbiome project started, they realized there were so many organisms that they actually talk about phyla. They don't talk about genus and species. They talk about phyla. They talk about big ideas and big groups of organisms. So the firmicutes or firmicutes refer to multiple gram-positive organisms that you probably are quite familiar with. And then the Bacteroidetes, as I said, the Bacteroides. And these are the most common phyla, these two, in the GI tract, much more than the ones that you typically think of in terms of gram-negatives and then other bacteria. And we know that, at least they have described that, the microbiome, not only is it made of these organisms, but it changes with aging. So something interesting, and I think that this is something that we'll talk about a little bit, but we know that when babies are born, they really have very few microorganisms in the GI tract, and they inherit a whole bunch of them from the mom during delivery. And then during puberty, adulthood, old age, relative numbers change and relative strains of organisms change. And there's been some interest in looking at what that means. Does that mean that if you have more of this type of organism, you're more likely to have long-term gastrointestinal issues as you age, or vice versa? So a lot of interest in that. I didn't want to use the word, I hate this word, where is it? Immune senescence. Like, who made up that word? Most people think that starts around 40. Oh, thank you. Okay, so I thought I'd put this up because by now you guys are all going, not sure if I like this talk, and it's nice and bright and yellow. So, but it gives you an idea of these different phyla, which is one of the concepts that they are using when they're talking about the human microbiome. And here's this firmicutes, and I'm not really sure how well you guys can see this, but you see that there are a lot of the gram-positive organisms that you're quite familiar with, including Clostridia, which we know are commonly found in the GI tract. And then the Bacteroidetes are up here, and then the Proteobacteria are the more commonly thought of gram-negatives that you might see in stool, like E. coli and things like that. And this is not a new concept, but we recognize that specific bacteria live in specific areas in the GI tract. And with this work in the human microbiome, they've also noted that there are changes in the microbiome with different diseases. And this is what I sort of alluded to a couple seconds ago, where if you look for it, you will find it. So if you want to know if there's changes in the human microbiome with inflammatory bowel disease, you can find things about that. But if you want to know if there's changes in the human microbiome with depression, you can find things about that. If you want to know if there's stuff about it with psoriasis, you can find things about that. So really, it's amazing the degree of interest and associations that this whole human microbiome thing has initiated. But changes with pulmonary disease, digestive disease, different cancers, obesity, coronary artery disease, all sorts of central nervous system things, changes in the gut microbiome, and this idea of dysbiosis, which is kind of another good new word. And like I said, this is not a new concept, but this was certainly emphasized with the work done with the Microbiome Project. But we know that depending on where you are in the gastrointestinal tract, you'll find different types of organisms. So in the stomach, we tend to find relatively few organisms. Helicobacter pylori is one of them, if you're not very lucky, but lots of gram-positives and yeast. And then as you travel into the small bowel, maybe a few more organisms, and it's not really till you get to the distal ileum that you start to see organisms in any significant number, and then in the colon, start to see many more anaerobes than you do aerobes, like E. coli, or something like that. And it's not just that different organisms inhabit these areas, but they, in theory, have different effects potentially on our health because of their different metabolic pathways. So this was something that's quite difficult for me to understand. It was very complicated, but they just tried to demonstrate that different organisms have different metabolic characteristics and potentially can affect health in different ways. So the gut microbiome is predominantly made of these 4 phyla of organisms, mostly the Firmicutes and the Bacteroidetes. It changes depending on where you are in the GI tract. It changes depending on whether or not you might have gastrointestinal illnesses, things like that. Okay, so what does that mean? Well, it implies that a healthy gut microbiome is essential for health. And again, there have been a lot of associations and papers written about associations of why that is and how that is. And some of it is fairly straightforward. You can imagine that if you have a good, healthy gut microbiome, you're less likely to have pathogen overgrowth, right? If you've got other organisms living there that are happy and healthy, then it's less likely that you'll have someone else kind of moving in and causing trouble. This is a little bit more difficult to understand, right? Because this kind of goes to the next step of, well, if you have more short-chain fatty acids around, that's going to alter the way that your intestinal epithelia metabolize. And then, as I said, all sorts of interesting things about endocrine function, neurologic signaling, etc. I should also mention metabolizing bile salts, different effects on drug metabolism. How about that? You know, we recognize all the time that different patients react differently to multiple different types of medications, and it's worth pausing and wondering, hey, why is that? Is it just have to do with their own metabolism, or does it potentially have something to do with their microbiome metabolism and the microbiome that inhabits their gut? One would wonder. By the way, this is a nice paper if anyone wants to sit and read a lot about the gut microbiome. I can give it to you later if you want. I can tell. Yes, you do. You do. And as I said, you can really, it's fairly easy to understand how the microbiome acts as host defense, either by direct inhibition or by helping to maintain the cellular barrier, maybe making a different milieu in terms of nutrient utilization. I mean, that's fairly straightforward. At least it's easy to conceptualize. But it's interesting to think about what kind of things can influence your gut microbiome composition. And again, there have been some really fascinating observational studies done looking at what happens to somebody who is born in sub-Saharan Africa and what does their gut microbiome look like. And then if that person moves to Texas, what happens to their gut microbiome and how might that affect how they metabolize food or other aspects of their health. So that's been looked at and is really quite interesting. We talked a little bit about age. So we know that as you age, things change, or do they ever? And then lifestyle and diet, I've sort of alluded to a little bit, but certainly having a higher fiber diet can lead to some changes in the gut microbiome. And then just different host genotype, certainly varies from person to person. And of course, antimicrobials. And if you think about it, there's not a lot of things that we can actually do much about or that are variable directly other than things like lifestyle and environment and whether or not you're exposed to antimicrobials. So, because this is what I do every day, I thought I should say, hey, what do we know about what antibiotics do to the GI tract microbiome? I mean, we all kind of have this vague idea of antibiotics kill stuff. Yep. Okay. Antibiotics kill stuff, and then sometimes people get C. diff. Okay. Some antibiotics are worse than others if they have particular activity against anaerobes, because we know that a large proportion of the organisms in the gut, particularly in the colon, are anaerobes. So if we use a drug that's very active against anaerobes, that might not be that great. Okay. But really, which organisms are we talking about and which antibiotics are we talking about and what happens when you do it and how long does it last and are we supposed to worry and do we have to worry all the time and what do all these things mean? So I'm going to show you a little bit about what people have demonstrated with qualitative and quantitative changes in the microbiome after antibiotics. And then, and this part's crazy, what kind of clinical associations and changes people have demonstrated and talked about with those changes in the microbiome with antibiotics. Did you follow me? A little bit? Okay. So what happens when you give someone antibiotics? What happens to their gut microbiome? If you look this up, this paper will come every single time. So this was a paper that was published in 2007. They looked at 4 people who volunteered to take clindamycin, and then 4 people who didn't take clindamycin, and then they sampled their bowel microbiome, both by stool samples and by, I think, colonoscopy, if I remember correctly. And then they looked at which organisms were there and how they changed and things like that. And they chose clindamycin specifically, just as we were chatting, because they knew that it had particular activity against anaerobes, and they really wanted to see what was happening in the lower GI tract. And surprise, surprise, they saw that, yes, if you give someone clindamycin for 7 days, it affects anaerobes, particularly Bacteroides, and this alteration can persist up to 2 years later. Does that mean something? I don't know. There was another paper, again, if you look this up, this one pops up every single time, where they gave 5 days of ciprofloxacin, and they looked at gut flora, and they looked at sort of both the diversity and the abundance of the flora, so not just which organisms were there, but the actual number of organisms that survived the onslaught of the ciprofloxacin. And they, in fact, saw that most recovered by 4 weeks. But some, again, specifically these ones on the Clostridiales, the abnormalities persisted for up to 6 months. And then if that person had to take ciprofloxacin again, then it perturbed their gut flora even more. Good word, hey? Perturbed them. And this is the kind of work that these people are doing. This is that same paper that I just described. And the little pink, I hope you can see it as pink, the little pink balls are the richness, diversity, and evenness. They're just descriptions of how the gut flora changes. So that's on ciprofloxacin, and then the little green triangles are what they're like before, and the little blue diamonds are what they're like after. So you can see that they for the most part kind of return to baseline, but not entirely. Okay, so you take antibiotics, it kills bacteria. I think you guys knew that before you came in, right? So far, so good? Right. But the question is, am I particularly worried about that? I mean, I've been worried about C. diff forever, that's fine, but are there other things that I might be doing inadvertently when I'm trying to treat my patient for paronychia that may or may not need to have antibiotics, right? Like, or I could maybe use Keflex instead of clavulin, which has more anaerobic coverage. So, you know, like, what does it matter? Why should I worry? The one association that I really want to focus on for a few minutes is how antibiotics and change in gut flora might be related to obesity, because it's fascinating. I'm not going to talk about a million other things that are super interesting as well, and the one that I would actually recommend that if you're interested in this at all, you spend some time looking at is antibiotics in childhood and development of allergies and asthma. And so I didn't want to pretend to even talk about that because I'm not Brett Finley, who, for those of you who don't know him, is a spectacular researcher that's centered out of UBC. I really thought I could talk about this, but I'm like, no, I can't. I can't talk about that because he's way better than I am. So I'm going to talk about antibiotics and obesity, but there are beautiful papers that he's done that focus on antibiotics and change in gut flora and asthma and allergy. And then I'm going to talk about antibiotics and C. diff. And then, like I promised, I really wanted to try and focus on, all right, if I'm doing all these things to gut flora while I'm using antibiotics in my patients, then can I do anything about it? What do we think about fecal transplant? What do we think about probiotics? Are you following me so far? I just taking you where my brain wandered. And this is where I went. So let's look at antibiotics and weight gain. So we've known for a long time that antibiotics have been used in animal husbandry, right? We know that the animal, I can't even think of the word right now, but they've been using antibiotics, especially things like related to vancomycin, penicillins, to help animals to grow bigger. And this is my fat cow, who is very, very fat. And so we've known that for some time, and it's been a big problem in terms of developing antibiotic resistance in animal husbandry, and that's been translated into humans. Anyways, that's another discussion. But did you know that if you took a germ-free mouse, again, did you know there were germ-free mice? Did you know that you could buy a mouse that has no bacteria in its GI tract? One person knew that. I did not know that. Okay, so if you took a germ-free mouse and you took some of the microbiome from an obese mouse and you put it in that mouse gets fat. But if you take the organisms from a slender mouse and you put it in that mouse does not get fat. So interesting. So what does that mean? Like, is there some way that you can alter the GI tract, at least of mice, that can make them be obese or not obese? Well, frighteningly, we may actually be able to do that. And more, there's been interest in, we're really good at making fat cows. It looks like we might be good at making fat kids too, which is a concerning thing. And there have been some papers done starting around sort of 5-7 years ago that have looked at what happens to children who are given antibiotics early in life, how that changes their gut microbiome, and how that puts them at risk for obesity. So again, I direct you to the amazing work that Brett Finley is doing. If you haven't read his book, Let Them Eat Dirt, have you guys ever seen that? It's awesome. I'm feeding my kids dirt every night for dinner now. It's awesome. You're supposed to get a dog and anyways, that's a whole other story. So all sorts of interesting things that he's doing where he's recognizing that a healthy gut microbiome that is exposed to as many sort of diverse organisms as early as possible and has as little antibiotic exposure as possible is associated with less common development of things like obesity and, as I said, asthma and different allergies. Really interesting papers. And one of the theories is that, and this is a theory, this is just published a few years ago, but this is a theory about how this might happen with antibiotic therapy. The healthy gut microbiota, it has effects on development of adipose tissue, muscle, bone, and also the immune system through T cells. And then if you change those organisms and you change, as you remember we were talking about it, which short-chain fatty acids you make, how many you make, how they affect your neuroendocrine system, things like that, then you may end up with alterations in how these different tissues develop and favor, which ones are developed to a greater degree and to a lesser degree. Now, so there's this concept that in mice, you can alter gut microbiome and make them fat or thin. Fine, those are mice. There's this concern that if we give antibiotics to animals, they get fat. If we give antibiotics to kids, especially when they're young, they're at greater risk of developing obesity. How about in adults, in grown-ups? What do we know about that? Also so interesting. Are you guys having fun? I'm having so much fun. Okay, so how about in adults? Well, this paper took 12 obese people, not very many, but you have to realize that when you do these studies, it's a lot of poo. There's a lot of samples that you have to collect and a lot of organisms that you have to look at and a lot of PCRing and all that kind of stuff that you have to look at. So it sounds like small numbers, but there are a lot of organisms. Anyway, some of them they put on a fat-restricted diet. Some of them they put on a carbohydrate-restricted diet. And then they looked at the gut microbiota over the course of a year. Like I said, lots of samples. And they did see changes in the relative number of organisms. Now, firmicutes are by far the most common no matter what, no matter if you are slender or not slender. But they did see changes in the number of organisms. And that was irrespective of what type of diet they were on. Because you could say to me, well, that's just because they ate lots of bananas, and then people that eat bananas get different organisms. But actually, it was irrespective of the type of diet that they were on. And you can see that the idea is that lean individuals have relatively more bacteroidetes than the obese individuals. And as these heavier individuals lost weight, they gained relatively more of the bacteroidetes. And there's even more kind of to it if you kind of go into it a little bit deeper. The theory is that the non-obese people and the weight loss people that increase their relative number of bacteroidetes, that focuses more then and has more effective and efficient polysaccharide metabolism, whereas the ones that have relatively more of the firmicutes have more sort of a little bit more towards lipid metabolism. And if you can get this, their poop has fewer calories left in it. So the people, and so the people that are more sort of on the bacteroidetes side, they're not as good as at absorbing all the potential energy in the poop. Because did you ever wonder, you've always had your best friend in college who ate everything, right? And then there was, your friend down the street who never ate anything, and you never really understood why that and why. Anyways, maybe this is one of the reasons. So you have different gut bacteria that's better or less able to absorb and break down carbohydrates. So what is yet unknown, and And I think everyone's going to start asking, and this is where I want you to start thinking about what does this mean to your patients and how can you help them approach this in an intellectual and helpful way. What does this mean in terms of therapeutic manipulations of the human gut microbiome? And does that mean that we could take a pill full of Bacteroidetes and lose weight? Well, nobody knows, and that sounds a bit crazy, so nobody knows. But I think the danger is, and what you'll see if you sort of troll through the internet, is that's what's out there. There's all sorts of little advertisements that will tell you, do this, increase your Bacteroidetes, lose weight. It's all kind of pseudoscience. What do we know about, like, what does work in terms of therapeutic manipulations of the gut microbiome? Well, there are some really nice papers that that come out in favor of fecal microbial transplant when treating C. diff. Now, I put this up just so I could draw your attention to the fact that there are new C. diff treatment guidelines that were just published last year in the Journal of the Association of Medical Microbiology of Infectious Disease Canada. And one of our ID people at VGH is one of the co-authors on this, Ted Steiner. And one of the options for patients with second or subsequent recurrences of C. diff is fecal transplant. There's also this vancomycin taper. If you ask me, well, couldn't you use fidaxomicin? Yes. I mean, there's other options too, but one of the options that they actually have in the paper is fecal transplant. And I just thought you should be aware of what kind of data there is to support fecal transplant, because it started off that really there wasn't a lot of data out there, but over time there have been increasing number of patients, and this particular paper had almost 175 people that had fecal transplant, and they had fecal transplant by enema, which is an advantage because sometimes they're sick and you don't really want to be doing colonoscopy on them, and they get success rates either after the first or second attempt at fecal transplant that range, around the 80 and 90%. So a lot of interest in that kind of manipulation of the GI tract, particularly in people who have recurrent C. diff. Now, why does it work? By this time I was getting silly. By this time I'm like, okay, poo emoji. I'm sorry. Okay. So why does it work? I mean, like, you sort of, again, you're like, okay, well, that makes sense. Let's put some healthy stool into another guy and see what happens. But don't you really want to know, like, what really happens, right? Oh, and then they get better, yay. But don't you want to know, like, what really happens? Like, what organisms are there and which ones aren't there and where did they go and how long do they stay there and will the C. diff come back? Like, don't you want to know those things? I did. So we don't really know yet. But if you look at the microbiome sort of pre and post fecal transplant, some of the studies are showing that persisting donor organisms are there for years post-transplant. And there have been these really pretty papers that try to look at, again, sort of donor microbiome diversity and different and organisms. And the donor ones in this little picture are these green ones that are healthy, and yay, green is good. And the people with recurrent C. diff, bad, red. And then the ones that were post-fecal transplant sort of had some overlap with the donor ones and some very interesting outcomes with respect to the specific organisms and the diversity of organisms and the dysbiosis. Okay, are you keeping up with me? Okay, so we talked about how the microbiome changes when we give people antibiotics. And then we talked about what does that mean? Well, we know about C. diff, fine, but there's increasing evidence that if you change the microbiome with antibiotics, you might put someone at risk for obesity. So if you put someone at risk for obesity, why is that? Why? Which organisms are different? And we realized that there's some evidence that if you have more Bacteroidetes versus Firmicutes, you might be more likely to be a lean individual rather than a heavy individual. Some really interesting work there and how that is tied into using antibiotics. And we talked about, well, what can we do therapeutically to affect the gut microbiome? And what do we know? What does work? What doesn't work? And if you have a pathologic state like C. diff, we know that using something like fecal microbial transplant can actually positively affect the human gut microbiome and lead to decreased disease. Is there anything else therapeutic that we know about that can alter the gut microbiome? Oh, and this is another wormhole, people. How about probiotics? Is there any evidence for use of probiotics? What are the potential risks and costs? You know, should we be using them? Don't your patients ask you every day? Mine actually, no, mine tell me, blah, 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 but I'm taking my probiotic. Okay. I didn't ask you to take your probiotic, but okay. So again, you'll be amazed at what is out there on the internet. This was my first one that made me kind of go, okay, 7 reasons to take probiotics. 1, boost your energy level. I'd like that. Increase your metabolism. Oh, yeah. Maybe there's bacteroidetes in there. Maybe that's why. Helps to detoxify my body. Okay, if it makes me, you know, maybe I'm better at metabolizing things and less toxins. I don't know what kind of toxins, but okay. Help to fight inflammation. I'm not sure about that. Helps to absorb minerals. Okay. Helps to fight food sensitivities and helps to keep insulin levels in control. They started off okay, but then they got pretty pseudoscience at the bottom. This one's awesome. Okay, the pros of probiotics. I'm not going to make you look at all of them. This is my favorite, Save Paper, because it reduces the risk of antibiotic-associated diarrhea. But anyways, I thought that was funny. And then, do you like this one? Improved levels of psychological distress, including depression, anger, hostility, anxiety, and problem solving. So all those things will get better if you take probiotics. Sounds perfect. And don't forget your kids. This actually disturbed me. You can go and buy probiotics specifically for kids, specifically for use in children 3 months and older, to mix with breast milk. Not sure if that's what God intended. Not sure. And this one, the best. Okay. What if a probiotic could help anxious dogs feel calmer? So they don't go as far as actually saying probiotics do cause, you know, your dog to feel calmer. This is my dog. My dog is actually trying to slowly kill me. She's 8 months old and not really sure why we got a puppy. And that's a whole other discussion. But she's very sweet. And that's after I just ran the snot out of her. Yeah. Okay. So what are probiotics? Well, they have to be live microorganisms, and you administer them in some kind of adequate amount that they're supposed to do something good to the host, like maybe deal with anger issues. Not sure. So just to give you some kind of feeling for what we're talking about, the global sales of probiotics are somewhere around 30 billion in the States, and that's like twice the PPIs. So if you think about how many people you know are taking PPIs and what a huge industry that is, probiotics is probably about twice that. It's the fastest growing segment of global, you know, dietary supplements and food industry supplements. But it's not easy to be a probiotic because really our bodies are designed to not have live organisms enter here and get all the way down there, right? So they have to survive the passage through the digestive system. They have to be able to attach to your intestinal epithelia. They have to have like a shelf life. That's not easy either. What are you going to do? Like, how do you package them to say, okay, stay alive there, stay, we'll die, right? So, and they have to be able to utilize whatever you're eating and metabolize whatever you're eating, and most importantly, be non-pathogenic. So, as you can see, not easy to be a probiotic. But there are some interesting theories about why they might be beneficial. And I'm not going to go through all of them, but, you know, just use common sense, right? I mean, if you have probiotics around and then you have some overgrowth of C. diff that's trying to get in and do something bad, like maybe the probiotics can actually kind of get in the way, and then those organisms can exclude the pathogenic organisms, or maybe they can alter your gut immunity so that you're more able to protect yourself against pathogenic organisms. I mean, it sort of there's some reasonable theory about why it might do that and why you might want to consider taking them. But is there any evidence? Well, I looked at this actually some years ago. This is the paper that was published in 2012. And I remember when I looked at it the first time, I kind of came away thinking, and I still feel like, I'm not sure. There is this meta-analysis that tried to see whether or not probiotics had any beneficial effect in protecting people against antibiotic-associated diarrhea. And they did come away with a relative risk reduction that's significant, with a number needed to treat of 13. The issue is that the papers had significant heterogeneity to them, not so much in like, was it a randomized trial, blah, 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 but the organisms that they used and whether or not these people had viable organisms and things like that. There was a lot of heterogeneity there. Different types of probiotics and dosages. The commonest species of probiotic studied was the Lactobacillus, and then there was also some people that looked at Saccharomyces, and they showed some promise in those organisms. But this study that was published in 2013, which had a good number of people, 159 events, with a total of almost 1,500 microbial preparations that people looked at. This one, in fact, did not show any preferential or positive effect from using Lactobacillus in either treating antibiotic or preventing antibiotic-associated diarrhea or C. diff. Now, these patients were older. These were all patients over the age of 65, so maybe it's a matter of it doesn't work in the elderly, but maybe it works in young people, but we just don't have those answers. So if you look for it, you'll find evidence to support the use of probiotics in antibiotic-associated diarrhea. Primarily, this study comes out saying, okay, maybe it's something worth considering, but we don't know a lot about which particular probiotic are the most useful, and there's this concern that perhaps there was too much heterogeneity in the paper, and then instead this trial, the PLACIDE trial, did not support their use. So not really clear about that. And then the question comes, are there any risks to the use of the probiotics? Because that's part B, right? Like, or maybe it's part A. It's really primum non nocere. We're not supposed to hurt people at the beginning, right? That's what physicians are supposed to do. So there's certainly a lot of description of GI intolerance. So if you look for it, you'll find it. There's some interest in using probiotics in irritable bowel syndrome, and depending on which paper you look at, some people get better, some people get worse, some people say the probiotics give them this, that, and the other. So certainly you can imagine that. You're putting live organisms in your gut. Maybe it's going to give you a little bit of bloating. I don't know. And then again, significant variations in preparations, and in some of the papers, 30% of tested products did not contain a single viable organism. So not sure how that would help, and hard to make that happen. And then there have been some reports of really significant adverse effects associated with probiotics, and the big ones that people are aware of, I think, are episodes of bacteremia or fungemia, primarily with Lactobacillus, and then this one was Saccharomyces. So it's not a matter of just having a little bit of a tummy ache if you take probiotics and you don't need them. It's really a matter of potentially being dangerous in some hosts. Now, this paper was just published last year, and this kind of takes it one step further. So it's not just a matter of, well, probiotics are expensive, and maybe they'll help you with antibiotic-associated diarrhea, maybe they won't. Doesn't really look like they protect against C. diff. Not really sure if you'll get viable organism or not. Lots of questions around that. But then the question is, what actually happens to your gut microbiome if you take probiotics with antibiotics and what happens to it if you don't. And this paper looked at 21 healthy adults, and they took ciprofloxacin and metronidazole, which, by the way, is kind of, one of our nuclear bomb attacks of antibiotics, right? Like, this is a pretty broad spectrum, aggressive antibiotic. And after the course of antibiotics, some of the participants took probiotics twice a day for 28 days. This was the other thing that made me surprised when I talk to patients. When they take the probiotics, and then they say, but I'm still taking them. I'm taking them for another month. I'm like, okay, who told you to do that? I didn't tell you to do that. Anyways, 6 of them received an autologous fecal transplant. These guys were not fooling around in this study. And then 7 had no further treatment, okay? So everyone took the antibiotic. Some of them stored some of their feces beforehand for fecal transplant later. Some people took probiotics, some people got nothing, and then they all got multiple samples, and they got samples not just of bowel movement, but also from the bowel itself. They had colonoscopy, right? And this was what they noted, was that post-antibiotics, probiotics actually delayed the gut microbiome reconstitution. So what they came away with was, we all try and return to our homeostasis. Sure, if you really look for it, probably, and I'm sure even if you look at these pictures, there are changes in the gut microbiome that persist after taking antibiotics. I mean, I've shown you that already, right? When in the clindamycin paper earlier on, I mean, there is an effect of antibiotics on the gut microbiome. But the question is taking probiotics going to make that better, or is that going to make that worse? Well, what they're trying to show in this is that this is the pattern of your gut microbiome off antibiotics. Okay, so these are some gram-negatives up here, and then multiple other organisms here. Then you take antibiotics, it changes. You kill these ones, don't kill these ones. And then with fecal microbial transplant, you reconstitute much of the microbiome spontaneously. You reconstitute, much of the microbiome. Wait! And then the probiotics, though, leave it messed up. Like, the probiotics continue to alter your gut microbiome. So, I'm not sure. When I started looking at it, I actually really wanted probiotics to be good. Don't you kind of really want them to work? Like, because part of me wants to be able to do something, right? Like, I'm giving the antibiotics to my patients. I know I'm wrecking their GI tract microbiome. I know I'm doing that. So part of me wants to kind of say, oh, it's going to be okay, though, because take these, and that will fix it. But after having read all this, I'm not sure that I can say that yet. I'm not terribly worried about them being dangerous in most of my patients. Most of my patients aren't leukemics who have enterocolitis. Like, most of my patients aren't those patients, thank God. I mean, most of the patients are being treated for skin and soft tissue infection, and they're otherwise relatively well, things like that. So I'm not terribly worried about those kind of things, but I don't feel super strongly that taking probiotics is really the way to go to protect the gut microbiome. So there you go. We just took a little wander through where we are with the gut microbiome. So what I'm hoping you'll take away from this is just a little bit of knowledge about what the Human Microbiome Project was, how that was done, focusing on the concept that most of the organisms that they've described are not culturable in the routine lab. They use very sophisticated molecular biology techniques to describe all sorts of organisms that we don't typically think about every day when we think about the GI tract and the human microbiome. And there's lots of ongoing study now about, well, if this is the healthy microbiome, what kind of microbiome do you have if you happen to have colon cancer or you happen to have ulcerative colitis, et cetera, et cetera? And is that a chicken or an egg? And does that affect the disease or not, right? And then what I'm hoping you'll think about a little bit is if there is a change in the microbiome associated with antibiotics, and we know there is, and I hope you remember a few little papers I showed you about that. What kind of things have we associated with those changes? And the one that I'm interested in personally myself is how is it affecting obesity, particularly in children? What does that mean? Why is that happening? And is it something that we really need to think about what we're doing and why we're doing it? Which brings us to stewardship. There. Hi. There. But much more needs to be explored there. And then I hope you think a little bit about, well, there are some therapeutic interventions that have been shown to work in affecting the gut microbiome. The most interesting one for me is fecal microbial transplant. But when it comes to probiotics, I'm not convinced that we're in a situation yet where we can come out heartily and endorse probiotics as something that can change the gut microbiome, except maybe for my dog. But that's all. Anyways, I'm going to stop there. I hope I can answer your questions. I don't know if I'll be able to, but I hope you found it interesting. Thanks.
Can I maybe just ask a question? It's very confusing for me that you initially say that when they define the microbiome, most of the organisms
are not culturable. Yeah,
they can't culture it.
Yeah.
So how do they decide then to make a probiotic if they don't even know what they need to replace?
I know. Well, that's a very good question.
So that's why I think we're not there yet.
We're very far.
To say, all right, we need to make this something that we can replace, but we.
Yeah, we don't even know what to replace. I totally agree with you. There's so much knowledge that we're still in the information gathering stage. And to try and jump from there to therapeutics, which is the danger. And that's, I think, what's happening, because of the internet, because of pseudoscience, because of that kind of thing. But it happens. I can't, if you actually spent 5 minutes and asked how many of your patients that you have on antibiotics are taking probiotics, you'll be shocked.
Yeah, most of them.
Right? Yeah.
Any other comments or questions? Yes, Keith.
Or not. Yeah, or not.
I really enjoyed that talk. You sound fairly positive about fecal microbial transplant.
Mm-hmm.
Are you doing that at all? And after your answer, I just have a follow-up.
Oh, okay. So I'm not doing it because no. But you can. But Ted Steiner at VGH is quite active doing fecal microbial transplant, and I've sent him patients, and he's quite willing to see them and moving forward there. Yeah.
My follow-up comment is Mark Wilkinson was a scientist here who's now in Spain who's doing this sort of stuff, and I don't think published yet. But one of his comments to me was that there's this thing that he calls up.
Jimmy, can we ask you to use the microphone?
And I'm trying to use the microphone. I'm sorry.
May we ask you to use the microphone?
It's just not working.
Sorry, not working.
The idea is you can check it anytime you like, but you can never leave. And what he means by that is it turns out you can go from a normal microbiota to one of these dangerous ones, a C. diff. Mm-hmm. And you can do it pretty easily with almost any choice of antibiotic. But once you get into that microbiome, in some instances, there is actually no way out.
To get out.
A course of vancomycin or something like that,
Yeah.
tapering, you will never get out of that California microbiome. And so a sort of a question is, should we be looking for evidence of that, the prolonged diarrhea, for example, and then go to a fecal microbial transplant?
So it was a comment first and then a question, and the comment was that Dr. Wally has a colleague that's doing some work on the microbiome and just was emphasizing the fact that once there's a pathologic alteration in the human GI tract microbiome, it's sometimes very difficult to reestablish the normal microbiome, even with antibiotics that you think might work, like an oral vancomycin for C. diff. And then the question that followed was, what do we do about that, really? Should we all be thinking about fecal transplant more positively, more aggressively, earlier in patients who are really struggling with trying to reestablish their normal microbiome? So maybe, yeah.
Yes, Gil, final question.
My question is, you mentioned very interesting talk about it. You mentioned that the infant or the fetus gets the microbiome during birth. So if somebody has a C-section, is it very different?
Yes. Such a good question. So, like I said, I had so much fun with this whole thing. And I just went, yay, no C-section. Okay, so the question was, if the infant acquires much of its GI tract microbiome and skin microbiome from the mother during a normal vaginal birth, what happens to the infant born by C-section? And actually, again, Brett Finley's group has done some really nice some interesting work on that, showing that the infants born by C-section, whether it's because of the fact that they don't get that passage through the perineum and don't get sort of covered in mom's microbiome, or whether it's the fact that they may have had some perioperative antibiotics, or maybe it's a combination of the two things. They have very different microbiome than infants that are born vaginally, and there's been some associations with greater risk of allergy, asthma, things like that. Yeah, and so there's actually people now that will do all sorts of crazy things. Again, internet. If they deliver a baby by C-section, they will inoculate it. Yeah. Yeah. With stuff.
That would be enough.
Yeah.
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 That's beyond the scope of
Okay.
the talk.
Okay.
Before thanking Dr. Montessori, I wanted to remind everybody next week is Respirology will be presenting Grand Rounds. Thank you for the great presentation. I love your dog.
Arlo. Her name is Arlo because my daughter doesn't prescribe to, gender, whatever. I don't remember what she said. It's like, well, it could be a girl's name or it could be a boy's name. Like, okay.
If I see Arlo on the tracks.
Okay.
Question.
Yeah.
The probiotics are always these capsules that you orally ingest.
Yeah.
Are there any rectally administered probiotics?
No.
Wouldn't that make more sense?
Yes.
Because you're going to kill everything that goes to the GI.
I know. Like, the only thing that
upper GI
makes it
tract.
through is, the odd lactobacillus.
So how can you,
And
why
they
would you
should be-
theoretically
and they
think
should be
you could restore it that
anaerobes
way?
too, right? And they should be anaerobes too, right?
Yeah.
So what do you do if you want to put something up there or in there, it should be an anaerobe,
Yeah.
right?
And whatever probiotic you take is not gonna have the biodiversity. It's gonna have one organism, maybe two organisms.
Waste, right?
Yeah.
And it's like so much money.
Yeah. So probably the way to go is if you far in the future,
Yeah.
if you know you're going to be put on antibiotics, you
Autologous stuff,
you collect a stool sample first,
Yeah.
freeze it or whatever,
Yeah.
hold it in good keeping in case you get C. diff.
Yep, that's right. You gotta do autologous. So good. That's awesome.
It's just the whole ickiness of collecting and storing your own stool. But,
Yeah, but that's
probably the way to go.
Yeah, you should have your own
Your own
supply. Yeah.
Your own staff.
Yeah.
Can I just ask a quick question?
Yeah.
Aside from the potential benefits in antibiotic-associated diarrhea, is there any evidence for any of the other claims on probiotics? Nothing.
Like anxiety and stuff like that.
Or
No.
even the other,
So yes. Okay. So with irritable bowel syndrome,
Okay.
there are some papers that come out in favor.
Okay.
So okay.
Okay.
And I want to say, in ulcerative colitis, it's depends on what you read, but not Crohn's, or Crohn's but not UC, one of one and not the other. And I want to say something about respiratory too. I kind of want to say asthma, but I can't remember.
Okay.
But yes, if you look for it, you can find things.
Okay.
Yeah, which are super interesting.
Yeah.
Totally.
Interesting, given that it's such a huge industry, as you said. A lot of people make money off of it. Yeah.
Right? More than PPIs, twice as much
Yeah.
as PPIs. Right? Oh my God. We are not doing we are in the wrong business, people. Right?
That was really interesting. Thank you very much.
You're welcome.
Oh,
Oh my God, he's back.
links all over my diet, starch, nightshade, now you have to
It's
go
like
and read for the
pre-science.
So he has one composer when you haven't heard of any other
Now I just mean it's
Yeah, good.
I know it's great. A lot of fun.
Compose yourself.
Yeah.
I want to talk about Montessori. And then teach you. When we see this, 30
Like it's all kind of the same.
Oh, I have no
But
idea.
at least you know there are some organisms there. Yes. Yes. But I still didn't find anything that was like strong.
Right.
Okay. Thing, but at least you know, you know, unlike me, on free to
These spread papers while antibiotics, and then it would be something that would be resistant to grow. So that's just my only caveat there. Is there anything else? Anything divergent? Yeah, we can just answer this. Okay. Tomorrow there's chocolate medicine around, something tonight. I'm just trying to figure out how to set up the conference room so we can all tune in. It's at VGH, but we can teleconference it. I just figured it's not worth starting today at VGH and like coming here, especially when we can teleconference. Oh, perfect. We're going to essentially get set up for us. Okay. So that's great. So just see it now.
I'm actually, you don't have a baby yet. When you have a baby, at 3 months, you feel like you're in the pit of despair and you will never crawl out. And that's
Not the best.
if you look at that, 8, you're like,
This is right. Yeah, if you go like that.
You should go like
it's
that.
still fun. I mean, it's still fun.
Okay, I'll see you guys tomorrow morning.
Oh, yeah. Oh, no, can I ask you one more? So he had his ultrasound done like March 30th or whatever, and then he saw surgery April 1st. Yeah, sound wise. Yeah. The whole thing is, I've never done it before. Like, so. Okay, so he had his ultrasound done. So April 1st, he saw one. I got a copy of the ultrasound. Yes, he was still in there. Yeah, no, so this one, like, March 30th, so it's like the day before he was and it's unchanged, just slightly bigger, but it's not an abscess, it's still like that. So, yeah, okay, he's doing fine. Discharge seasonal types. Discharge, but you know what I mean? Yeah. So he's off therapy. No, he didn't. So I was sort of thinking, I don't know if you're in next week or Natasha or who, but if one of us is in clinic next week or the week after, we should just at least sit down. I don't know. I'm just a bit nervous about it. I've never managed it before. I've never seen it before. Well, he is, but he was kind of like, it'll happen sometime. Oh, good. Okay. Yeah, 5 months on. Yeah. And he has been on so long. Probably we find out.
Oh, yeah, I thought you were in New
I've
York.
never been before. Missed actually. Oh, okay.
We're going to set it
Yeah.
up.
Oh, it's okay. Right? This makes me feel the same. I'm like, what is that? What is that? I'm like, boy,
And then
Are you still on that? Oh, okay, if you don't mind. Okay.
But also when you go away, just check it. I know last time I tried it, but I can stay. I love dogs.
Very good. She's a very good dog.
I was looking at dogs
so
today.
Close to like saying.
But you know what? Because my lab was a family dog too, but he was super hyper. We brought him to training. Failed. Well, I don't know if it was he failed because we took him out, because I felt like the teacher, like the instructor was. Yeah. So we know my dog, so then we took him out, so they never passed the course. But then he was crazy throwing his butt into the front. Also. You're on.
Yeah. I'm really like I'm this close. Well, 19, but he's not home. Yeah. And then 15, and that's we got the dog for her.
Yeah.
But she's like, out with her friends.
Exactly.
Which is great. Like, what do we? I have to stay on my mom and dad. Right? Like, I'm not that person.
Yeah. They
And
want
so,
the romantic emotions of the dog, not the work associated with the dog. So when we got cats.
yeah. If I'm not on service, it's totally fine. But if I'm on service, it's like, walk the dog for an hour, and do dinner, and do homework for the kids, and stuff for do the homework with the kids. Yeah. Sometimes I can't slip. Yeah. Blah blah blah, right? Like it's crazy. I can't do that. What the hell did they say?
He's fine.
I know.
Your call will be disconnected.